I will begin with looking at the biotechnology per se, in the sense asking a very basic question: what is biotechnology? And how it has evolved over a period of time. Some may say that biotechnology is a very new field, but the kind of facts I'm going to share with you will uh, definitely amaze you that human beings, with their evolution, had been utilizing natural resources from time immemorial. And in the process, the way they have used these organisms for their benefit, knowingly or unknowingly, have laid down the basis of today's biotechnology. Okay, so let us begin with, as I mentioned, asking a very basic question about what is biotechnology. Now, this word had been defined differently by different people, and if you look at the literature, you will see many definitions. So I have tried to put in here one such definition, which tries to explain what is biotechnology. So basically, biotechnology deals with manipulation of living organisms or parts of living organisms to make products useful to humans. Now, when I say manipulation of living organisms, it includes all the living organism spectrum, that is starting from microbes, plants, and animals. So we are talking about here manipulating all three forms of life, that is microbes, animals, and plants. And as you know, these living forms, some of them you can see by naked eye, and some of them you cannot. Okay, so including all those organisms, this field try to focus on manipulating the living organisms or parts of the living organisms to make products. Here, I would like to include one more term, that is services. So to make products or products which can be directly beneficial to the humans, or they may be used for to provide services. And I will explain what I mean by services later. Now, how that is achieved and what kind of manipulation one is talking about. So basically, this manipulation deals with the genes of organisms. Now, genes of the organisms are altered, which are responsible for their changed behavior, characteristics, and value. So basically, in order to manipulate organisms, we try to manipulate the genes in those organisms which alter their behavior, uh, characteristics, and the value. Okay. And lastly, basically, it is cell and gene technology used to produce new characteristics in plants, animals, and microbes, that is bacteria. So therefore, it becomes very important for us to understand what is this about the cell, whether it is a microbial cell, animal cell, or plant cell. And what is this gene we are referring to? Basically, as you know, each the basis of each characteristic of a living organism is associated with a gene. So if we understand the linkage between gene and the behavior of the organism very clearly, that how the genes are controlling the behavior of an organism or characteristic, then we can alter that gene and manipulate the behavior of the organism. Okay, and therefore it is very important to be very clear about the cell function, and then how various characteristics or how the genes are linked with the phenotype. So this linkage between gene and phenotype understanding is very, very important and significant for the field of biotechnology. Okay, now let us explore the let us explore the um, timeline for biotechnology. Hmm? How biotechnology has evolved over a period of time. You know, so as I mentioned, ever since the human civilization started evolving, the use of natural resources uh, also started. And as you see here, it dates back to 8000 BCE. And what was that activity? That is domestication of plants and animals. And what I mean by domestication of plants and animals is that the humans at that point of time started selecting plants growing around themselves and animals, looking at them and their usefulness. So they started picking up those natural resources, whether it is plant or animal, and then started using them and propagating them. And that is what is meant by domestication of animals and plants. So this phenomenon started almost 8000 BCE. And then you see a very significant development around 4000 BCE in Egypt. They developed, Egyptians, they developed art of making wine. Now, wines are made even today, but that would have been a very, very crude way of making wines at that point of time, because now we know the entire biology or molecular biology of this process. But then this process of making wine is not new. It is as old as 4000 BCE, you know. And then bread you eat, we all of us eat every day, and you feel that bread is the technology has evolved now. But then if you see here, uh, the Egyptians again used yeast to make bread, you know. So the technology is age old. It's not new. But then again, the advancement in this, the refining and defining this process happened much, much later. But use of biological systems to make products is not new. And that is what I'm trying to bring to your attention, you know. Uh, And then also around the same time, again, Egyptians, they also learn along with brewing, also the cheese making. All of you know that, cheese you eat every day. But then the art of making cheese is also pretty old, around 2000 BCE, you know. And then you have soybean curds. So that technology around 500 BCE, people had been using the soybean curds for treatment purposes, for treating boils, for example. And very interestingly, if you see around 300 BC, here, The Greeks, they developed grafting techniques. Now, you are familiar with grafting techniques in plants. You know, you can take a cutting from one plant and graft it to the other. So this again approach is not new. It's the Greeks started or developed this technique around 300 BCE. You know, uh, and then powdering the natural products and using them for different purposes started 100 CE, where they uh, developed a technique with chrysanthemum. They dried it and powdered it and used it for different purposes. And as the time went by, you see a very significant development uh, in 1663. I'm sure you have heard this name, Robert Hooke. Now, he described the cell for the first time. That is, that living forms, which you can see, plants or animals, they are made up of cells. Now, this concept was floated as early as 1663 by Robert Hooke, you know. And then around 1635, in the same century, Leo Hooke, he discovers the living forms which you cannot see by naked eye, that is protozoa, I mean the lower animals and the bacteria. And that could be possible with the development of the technology or techniques by which they could see, that is the magnifying techniques by the handheld lenses or simple microscopes of that time. And then see around 1997, the first vaccine came against cowpox. You know, so that was the beginning of developing technologies using biological systems or organisms. As you know, the basis of vaccine is at that point of time is the heat-killed whole organism. That kind of vaccine was first developed in 1797, you know. And then in 1800, the concept of biology first appeared, that biology is life, you know. So this discipline started as early as in 1800. And then in the same century, like around 1830, proteins were discovered, that the living forms, one of the constituent is proteins. So this discovery was made in 1830, in 19th century, you know. And then the first bacteria which was characterized and discovered was E. coli in the 1855, and followed by the discovery of fermentation, germ theory, that you can ferment an organic solution by inoculating with some microbes. So fermentation technology was discovered again in 1857. And then, see, the 18th century was very, 19th century was very rich in discovering biological systems. And it is here that Charles Darwin published his theory of evolution by natural selection, you know. And a few years later, we have Louis Pasteur, who developed the pasteurization process, which is being used even today for preserving biological system, and it is being used, you know. So, and then the classical understanding of Mendel's law of inheritance, 1865, which was discovered. I mean, the experiments, he started doing experiments in 1855. 10 years work he published in 1865 and gave the law of inheritance, which later on became the basis of genetics. And even today, these laws are, followed. And of course, they are validated, revalidated in the last century for all living beings, animals and plants. So all the living organisms follow simple principles of Mendel's to inherit characters from one generation to another generation. And then again in continuation in 1888, the concept of chromosome was developed. That is, the genetic material is located in nucleus, and in nucleus there are thread-like structure, what we call chromosomes. Okay, so even this concept was thrown in 1888. Coming to last century, which is the most productive time for biological sciences. So in the beginning of last century, the bacteriophage were discovered. Now, what is this bacteriophage? Bacteriophage, are the viruses which feed on bacteria. So even they were discovered. Okay, and the biotechnology in principle was introduced in 1919 by Karol Irki. So the terminology of biotechnology is not very new. It's almost 100-year old if you see from today's timeline. But then the understanding of biotechnology of last century of 1919 has changed completely, in the sense that now we know, if we talk about altering the characteristic of an organism, we know what we want to change and what is the basis of that characteristic and how we want to change everything. We have understood, although there are many gaps left even today, we cannot achieve what we want because our understanding about these processes is not very complete, and we need to understand, and I will come to these questions later, where we have reached and what are the lacunas of today. I will tell you as we go along during the course. But the point I'm trying to make here is that the biotechnology is not new. It is 100-year-old discipline, but then our understanding has changed with the knowledge we have gained about the cell and its function, the characteristics of an organism, the basis of that characteristic, and how that characteristic can be changed at will. That is the key to the biotechnology. Then, see, in 1922, the major discovery of a bioproduct, that is insulin. The insulin metabolism is the key to the most popular disease, diabetes. So this was discovered and understood as early as 1922. And then very important discovery in the last century made was Hermann Müller, who first demonstrated that radiations, the gamma radiations, UV radiations, they affect the chromosomes and they cause defects in chromosome. When I say defects in chromosome, as you know, these radiations are capable of creating mutations. And these mutations in turn are responsible for so many diseases, Now, again, And last century, the antibiotics were discovered, by Alexander Fleming. He was the first one to invent the first antibiotic, penicillin. And thereafter we have uh, so many antibiotics, the broad spectrum antibiotics, the specific antibiotics today in market, which are used for curing various infections because of bacteria. You and then the very key discovery, in this, if we see in 1944, where Avery and his colleagues, McCarthy, they first demonstrated that DNA is the one which carries the genetic information. When I say genetic information, I'm talking about the traits which gets inherited from one generation to another. That is because of the genetic material. The genetic material is in chromosome, which resides in nucleus of the cell. And in chromosome, they discovered, Avery and McCarthy and Leonard, that it is the DNA, which is the constituent of chromosome, or one part of the chromosome, which is which carries the genetic information from one generation to another. So that was the breakthrough, you can say, which is the basis of biotechnology. And then all of you have heard this work of Watson and Crick and Wilkins. They are the one who first gave 3D model of DNA, or they gave that how DNA organizes itself, in the cell, in the nucleus, in the chromosome, in 1953. And you are aware they got Nobel Prize for this. In fact, some of the names, or most of the names which I have mentioned in their work, they have been recognized for these breakthroughs, and they have won Nobel Prizes for their discoveries. And then in 1966, I'm sure you are familiar with the genetic code, how the DNA is cracked actually, what DNA contains, and what kind of information the DNA has. This is in the form of genetic code, and these are the people. We have Harkoven Krona, who was one of the co-discoverers of this genetic code, and all of them got Nobel Prize for their discoveries. And then, in around 1971, the first artificial gene was synthesized based on the sequence known. Okay, so that is again that it means that you can synthesize a gene outside the cell or in vitro, you know. And in around 1973, the Cohen and Boyer, they perfected the techniques related to genetic engineering. Means taking a gene from one source and putting it into another source. How that is achieved, that was actually perfected by these two guys in around 1973. So genetic engineering is almost 50-year-old concept and technique. Of course, that is being refined and defined in the last 50 years, though they demonstrated it in model systems, but then, using them, or using these techniques to all the living systems, plants, animals, microbes, they required sort of perfecting them, changing them, so that they can be applied for a particular organism. So that has been happening in the last 50 years, you know. So now if we say today, it is possible to engineer all sorts of organisms. You can engineer microbes, you can engineer plants, you can engineer animals. When I say engineer, I'm talking about we can change the behavior of an organism or part of an organism at will. And for that, we have to have the gene which is responsible for a behavior. Two, the technique by which you can take out the gene and then push it into another cell type. You know, so the technique has to be there, and that technology. What is technology in biotechnology? Is basically the gene which is responsible for a particular trait or a phenotype or a character. So this kind of linkage between the gene and the character, phenotype, and trait is very, very important as the key for biotechnology and genetic engineering. You know, so that started in 1973, and as I mentioned to you, in the last 50 years, this has been perfected, and we have examples from all over the spectrum of microorganisms. I mean, the organisms in general, whether micro, plants, or animals. And again, in 70s, if you see, a lot of exciting discoveries were made as far as biology is concerned, and production of monoclonal antibodies. Another one means you can generate antibody for a specific protein. You know, so I mean, that is the one which what we call monoclonal antibody means a particular cell, human cell, which will make only one kind of, I mean, it will predominantly make one kind of protein, which is antibody against an antigen, you know. So that technique was given in 1975, you know. And as I mentioned, insulin was discovered long ago in previous century, but then the first human insulin was produced in 1982, and that was marketed. And again, how the insulin is produced is again very interesting to mention here. You know, in good old days, the insulin was made for human consumption by slaughtering pigs and sacrifice and isolating it from their liver, you know. But now you can make insulin uh, in bacteria, and that is what is achieved here by engineering macro in such a way that it makes insulin, which is consumed by humans today. You know, so it is again in 80s, early of the last century. You know, genetic engineering, when you alter the behavior or trait or phenotype of an organism, we call it as transgenic, whether it is transgenic bacteria, transgenic animal, or transgenic plant. You know, so that is genetically engineering or genetically modifying an organism. So genetically modified organism, we call it as transgenic, plant, animal, or microbe. So successful demonstration or production of first transgenic animal was shown in 1981, that they could genetically modify. It means by then they had all the techniques in hand to achieve this. You know. And then the same decade, in 1983, a very uh, key technique was discovered, what we use very frequently in molecular biology or biotechnology is the PCR or the polymerase chain reaction technique. So this was also discovered uh, in 80s of last century. We will talk about this technique later, that what this technique is and what you can achieve with this. But at this point of time, I'm just trying to discuss with you some of the milestone discoveries and observations made, which have made today's biotechnology possible, starting from good old days of domesticating natural resources to this, where I have brought you, like the significant techniques like PCR, which was invented to understand the behavior of DNA, RNA in the cell, and which are linked with the characters and the traits. So utilizing all these things, again, the first recombinant vaccine was produced in 1986, which deals with the hepatitis B virus. Now, as I mentioned, vaccines have two form. One is you use the whole or a heat-killed whole organism and use that as a vaccine. And today, what we do, we try to find out the immunogenic regions of the proteins which interact with the host and use only those proteins um, which can which can induce the immune response in humans, for example, and that we call as recombinant vaccines. So it's not the whole organism, but only a protein, and nowadays we can make multiple proteins put together and use that as a vaccine. So the first beginning of recombinant vaccine started in 1986. And then around the same time, very important discovery in this is the anti-cancer drug, that is interferon. So even the first drug therapy for cancers was discovered in 1986 in the form of interferon. Now, all this, why I'm telling is resulted from the basic understanding about the biological system. Now, interferon came as a byproduct of understanding how the cell divides itself uh, within the organism or outside the organism, and how interferons can interfere that division, because cancer is nothing but the uncontrolled cell division wherever it starts happening, where it should not. I mean, when a cell starts dividing where it should not divide, it leads to cancer. So the first drug therapy came in 1986. Now that could be possible only by understanding that how cell divides. That's the basic question. So if you understand how cell divides, then you can control the cell division also. Okay. So therefore, it is very, very important to understand the behavior of the cell in general. And cell is the basis of life, from unicellular to multicellular, multi-organ organism, and therefore it becomes a key point to understand. Coming to other discoveries. Now, this is something very, very important from the plant biotechnology point of view, and again, it is around 87 or in 80s. The first GMO, or genetically modified plant, was produced. That is tomato, which was virus resistant. As you know, the plants are also sensitive to biological factors. So the biofactors means, like humans, the plants also get infected by viruses, bacteria, fungus. And when they infect plants or animals, this infection leads to diseases, and the disease leads to the death of organism. So, with context to plants, it is it is the first successful application of genetic engineering principles to modify the character of a plant, that in this case it's the tomato fruit, uh, uh, which was protected from viruses. I mean, the plant was protected from the viruses, and the crop could yield more and more tomatoes. And the first product from this GMO or transgenic tomato in the form of puree, tomato puree, you must be eating every day from market, it is available. But then it is for the first time in 94 that the first product from this genetically modified tomato was marketed in the form of tomato puree in 1994 in US, you know. So it's the application of biotechnology to improve a plant against the viral infection, and then using those products to make further products which are utilized by humans. Okay, so that way, biotechnology has changed the trait of a plant or behavior of a plant, and ultimately leading to products which are consumed by humans. And then this, if you remember, very popular and very much talked about case of Dolly, that is the clone. So in 97 of the last century, 90s, this is what was demonstrated, that it is possible to clone animals. And in other words, it is possible to clone humans. But since there are ethical issues involved, this was not translated to humans, but Dolly uh, is an example where animals can be propagated clonally. It means you can create a replica of a individual genetically similar, because whenever an organism undergoing sexual reproduction, it brings about genetic variation. They change genetically, but by clonally propagating, it is possible that you can generate another individual which will be genetically similar to this, and that is what happens in clonal propagation. You know, so it was demonstrated. It was demonstrated for plants that it is possible to clonally propagate plants to have the genetic similarity, but it was not possible for animals until the Dolly experiments were done and demonstrated. Okay, and then similar to this, the people working in another direction, they established the embryonic stem cell lines. You know, and what is the stem cell lines? You know, these are the cells which are capable of differentiating into as per the requirement to different cell types. You know, so this was demonstrated that embryonic cell lines is a stock of cell lines which can, if given the right kind of environment, can be differentiated into any specific cell type. Example, if you want to understand this, is like during normal development of animals, you know, the cell, the fertilized egg undergoes multiple divisions. And then, I mean, the first stage of development is the cell division, and then the differentiation, and the commitment on the part of the cells to give rise to a specific cell type to make a multi-organ, multi-cell organism. Okay. Now, and in nature, when the cell gets differentiated, it cannot revert back to normal. It will remain in that differentiated state. But the stem cells, particularly embryonic stem cells, they are capable of committing themselves to different directions of development, you know, and it is very, very significant discovery, and it is being utilized for therapeutic purposes for variety of diseases, and we'll talk about it in the other animal biotechnology part. And now here it is again a breakthrough of sequencing, gene sequencing, from gene sequencing to genome sequencing. And that's where the first genome was sequenced successfully. That is the Human Genome Project. And the draft version of this sequence was published in 2002 in Nature, actually. So it means what it means. It means that they had end-to-end sequence of the genetic material DNA, which constitute our genome. Okay, and that was something which is the information required. That what is, to begin with, what is the DNA content of the living organism, and in DNA, what is the sequence, and the third is what this sequence means to the organism. So, first step was to sequence the genome. So, sequence the DNA entire. Now, like human genome consists of 46 chromosomes, and on 46 chromosomes, you have DNA loaded, and that DNA was sequenced from all 46 chromosomes, aligned and resolved. Okay. Next question was to decipher this information, which is stored or present in the form of base sequences. You know, it is ATGC, that's all, which is responsible for holding or the genetic information. Okay. So, but then the significant part is the success story of sequencing the human genome. So this was the beginning, actually, and then in last 2 decades, this kind of effort have led to sequencing a large number of plants, large number of animals, you know, and looking at their genome and what this contains, and what are the similarities between these genomes, animals, plants, microbes, because that is very, very important step to understand what is the genome, what it contains, and how this genome functions. And that is the crux of biotechnology. If you understand these things, then you are done, actually. And the whole efforts of today is to understand this, the complexities of the genome, wherever it is, whether it is in simple forms of viruses, bacteria, and plants or animals. And then in the same thing, you are listening to COVID-19, you know, and if you have heard that a similar virus came in 2003, which is COVID-1 or SARS-1, actually. And at that point of time, this virus was also was a threat, but fortunately, this was not very infective. Although it also became epidemic, but then it was restricted. It could not spread the way the COVID-2 is spread of today. But then the point I'm trying to make here is that its genome was sequenced, in fact, and a lot of understanding was developed about this virus, which they are utilizing even today for with reference to COVID-2 or COVID-19. What are the differences in the COVID-1 and COVID-2? You know, so a lot of work was done with COVID-1, and a lot of information was generated, in fact. And then in the same story of cloning animals, the success story was with making a clone of a cat, which is a pet animal, you know. So, yeah. And then the development of vaccine, recombinant vaccine, which is again HPV1, and this is again HPV, and the total virus, and malaria-resistant mosquitoes, you know, in 2010, you know. And okay, so uh, to summarize what actually I had been talking and sharing with you is the use of natural resources in the good old days, and from there, as the knowledge or the development of technologies to look at biological system, to understand biological system evolved, uh, we started refining and defining these processes which were utilized earlier uh, based on the experiences, what's going on. They didn't know what is the science behind it, but now we know or trying to know wherever we lack the information or knowledge that what is the basis of, I mean, the scientific basis of that process which is being utilized, and also developing newer and newer, application-oriented utilization of biological principles, which can produce or give products which humans can utilize for food and health and even other areas as well. Okay. So, so to say that biotechnology basically is a good old days discipline which has evolved over a period of time, and the kind of biotechnology today we talk was not uh, existing at that point of time. Okay. Now, if we see recent breakthroughs in biotechnology, if we see, so we have improved food products for their nutritional values. You know, now we have, for example, rice, which can make vitamins, vitamin A, which is not made in rice naturally, but it has been improved or genetically modified. We will talk about this. And then we have targeted therapies for cancers, for example. We have known and developed technologies which can very targetedly send to the cancer cells to kill them. Okay. And also we have gene therapies wherever we have genetic defects. It is possible now to to it is possible now to treat. And then the CRISPR technology, which is very, very recent. This is the one which you can use for treatment purposes and also generating basic knowledge in molecular biology. Okay. So, to come back to what I have talked about is a broad area of biotechnology, uh, be it animal, plants, bacteria. But in today's context, actually, this biotechnology has been classified into different areas because the knowledge is being generated so much that it is very difficult for one to understand uh, under one umbrella, you know. So, for example, now we have microbial biotechnology, agriculture biotechnology, animal biotechnology, forensic biotechnology, bioremediation, aquatic biotechnology, and medical biotechnology. This is the list of the major areas. So, what happens when you classify or make different groups? It means we have now information so much information generated in under each one of these hats that they are made an independent identity. Okay, so for example, uh, what is happening in microbial technology or biotechnology? Here we try to manipulate microbes, whether it is bacteria or yeast. Now, what we manipulate? We manipulate them making enzymes, which are commercially utilized. So instead of extracting it from the source, you can make it here in the microbes and isolate from here, okay? Which are as good as or better than the one which one uses from the source, okay? And then you can manipulate these microorganisms to process the industrial waste, which are pollutants basically. Uh, and then you can make large quantities of proteins which are made in animals or plants. You can make them in microbes and then use them as a source to purify, you know. And these proteins are used or which are enzymes or otherwise useful products. You know. So microbial biotechnology deals with this kind of questions. If we look at agriculture biotechnology or what we call plant biotechnology, you know, here again the the plant productivity depends on the biotic and the abiotic factors. Biotic factors means the pathogens like bacteria, virus, fungus. You know, all these pathogens infect plants and reduce their productivity. Abiotic factors like the availability of water, high salt, and the drought and the heavy rain, all these factors affect the productivity. So we can use biotechnological principles to alter or manipulate the plants to withstand or to make them resistant to pathogens and tolerant to abiotic stresses, for example. And also we can improve the quality of the produce. We can make the kind of protein we want, or the how much carbohydrate should be there in the in the edible part, grains of foods, or the oil quality we can control by genetically modifying them. And also, we can make drugs inside plants. And the one I'm referring to is the possibility of making vaccines in plants. So, in other words, there is a time to come where you can be vaccinated just by eating few bananas or tomatoes, you know. So that kind of thing is possible. And we are going to talk in detail about the agriculture biotechnology or plant biotechnology. Okay. Animal biotechnology again as source of medical valuable proteins, antibiotics, transgenic animals, and basically they provide model systems to understand basic mechanisms, basic behavior of animal cells, you know, and generate information, and which can be applied to humans. Or anything before we take it to humans is tested on animals, you know. So, and also under the model systems basically. Okay. And the biotechnology has also helped in criminal investigations, like the forensic side. That is, that the criminal side, they can reaffirm or reascertain who has done the crime by simply looking at fingerprinting, DNA fingerprinting, you know. So this has been very extensively used for in the forensic side. Also, the biological principles and the tools and techniques, you know, which have been. And bioremediation is the same, which I talked about, developing microbes which can treat the industrial effluent, which are polycatted. There are so many success stories, you know, so far done. You know, for example, growth of bacteria, which degrades components in crude oils, you know, 1989. You know, this is the classical example of Exxon Valdez oil spill in Alaska and Deepwater Horizon spill. So basically, the microbes were generated to take care of this oil spills in ocean, you know, or anywhere basically. So in fact, these days, what is done is whatever industry which is generating waste that needs to be treated first by a technology which involves microbes actually to remove the toxic effluents from the waste, and then it is released to the environment. So that's a very very important part. And then the medical biotechnology, uh, which involves in diagnostics, treatments, preventive medicines, um, and then the gene therapies, stem cell technology being utilized, you know. So a whole lot of things are happening in this area as well, which you will be um taught by Shailendra Singh, who will be dealing with you animal biotechnology portion, which will definitely have input from medical biotechnology. Okay, so to summarize what I have basically um, taught is to give you a bird eye view about biotechnology, its timeline, how the humans have started or utilizing natural resources for their own benefit. Two, uh, today, how we are trying to modify and manipulate biological systems for generating products and services for the benefit of humankind. So, now